SwayamPrabha. Digital India. Educated India.
Dear students, you are welcome in this class. I am Dr. Dinesh Kumar, Principal Scientist from Division of Agronomy, ICAR, Indian Agricultural Research Institute, Pusa Campus, New Delhi. This lecture pertains to the course Agronomy of Oilseed, Fiber and Sugar Crop. The title of this lecture is Insect Pest and Disease Management in Crop and Processing of Sugarcane. Learning objectives are to describe the management of insect pests and diseases of sugarcane, and also we will explain you about the processing of sugarcane juice, how one can produce sugar and what are the major byproducts in sugarcane industry. Now you can see this is the plant of sugarcane and some glossary. Chlorpyrifos. It is an organophosphate insecticide commonly used to control a variety of insect pests. Fermentation, a metabolic process that converts sugar to acids, gases or alcohol in the absence of oxygen. Grub, the larval stage of various beetles found in soil and feeding on plant roots. So these grubs are also important pests in sugarcane. For example, white grub. We will discuss in detail. Now, honeydew, secretion produced by sap-sucking insects such as aphids, scale insects and white flies on plants attacking black moths. It is called as honeydew. So, honeydew is not good for the plant. So, these certain pests, they release the honeydew, which is kind of excreta on the leaves of the plant, and then sooty mold grows on it, black color fungus, and which restrict the photosynthesis by the leaves. Photoplasmas. Phytoplasmas are obligate intracellular bacteria, live and multiply inside living cells of host plants, causing diseases or insect vectors. So, these phytoplasmas are also important. Phyto, P-H-Y-T-O, phytoplasma. Ratooning, practice of harvesting a crop but leaving root system and part of above ground stem intact so that the plant can regrow and produce new crop without replanting. So, in case of sugarcane ratooning, the main crop or planted crop or first crop is harvested. After harvesting from the surface or ground surface, the stubbles are left. These stubbles can regrow into new plants, which is called as ratoon. Sooty mold, fungal disease that grows on plants having honeydew. It is a sticky substance of excreted by some insects, does not infect plants but grows on plant parts. Now, let us see insect pests and their management. So, sugarcane is basically long duration crop, therefore, it is attracting number of insect pests from planting up to harvest. So far, 288 insect pests have been recorded to infest sugarcane and about a dozen have been recognized as major pests. Damage during germination and tilling causes yield reduction, whereas during grand growth phase and ripening mars or reduces juice quality and sugar yield. We will see detailed symptoms or infestation, what happens to the plant, how they are doing damage, means nature of damage and how these insect pests can be controlled. We will see one by one. So, first one is early shoot borer, or simply it can be called as shoot borer, as the name suggests that it comes at early stage. So, scientific name is Chiloinfuscatellus. It infests shoots till 4 months after shoot emergence in subtropics. Larvae bore through the successive sheath and reach growing point, causing dead hearts being easily pulled out. So, you can see this is the larvae of the early shoot borer. You can identify. And now these are the dead heart symptoms caused by early shoot borer. You can see the central leaf which is which has just opened, it has turned brown. So, this is typical symptom of early shoot borer infestation. You can see dead hearts on the right side also. Removal of infested plants, trash mulching and light earthing up reduces incidence. Means if we are managing this insect, we can start with trash mulching and light earthing up reducing incidence. Soil application of chlorpyrifos 20 EC at the rate of 1.5 liter per hectare or fipronil 0.3 G at the rate of 25 kg per hectare at planting. Either of these insecticide, chlorpyrifos or fipronil, can be used to control early shoot borer. 4 sprays of granulosis virus of shoot borer with a dose of 10 to the power 7 to 10 to the power 9 IB per ml effective in tropical India. So, biological control method is also available for early shoot borer insect. Number 2 insect is root borer. Scientific name is Emalocera depressella. Formation of undetectable dead heart and a hole at shoot base. Apply chlorpyrifos 20 EC at the rate of 1.5 liter per hectare. This quantity of chlorpyrifos can be dissolved in 600 to 800 liters of water and sprayed per hectare. And bioagents are also available, like release of Trichogramma chilonis at the rate of 50,000 adults per hectare from July to September at 15 days interval can control the root borer. Third insect is top borer or top shoot borer. You can call it by either of these two names. So, it is Sarcophaga excerptalis. Larvae infest crop at all growth stages with reddish brown. You can see on the right side the infestation symptoms. Reddish brown, charred and sometimes curved dead hearts. Leaves with short holes and galleys in midrib of leaves. In grown-up canes, top internode buds give rise to side shoots forming a bunchy top. You can see short holes on the leaves. You can see more symptoms. It can also cause dead heart. And you can see bore holes. Holes may be there here. And you can see bunchy top. The growth is suppressed. What can be management for top borer? Collection of egg mass laid on underside of leaves covered with buff or orange colored hairs and removal of infested plants periodically. Soil application of carbafuran 3 Cg at the rate of 33 kg per hectare or chlorantraniliprole 18.5 SC. SC is your soluble concentrate at the rate of 375 ml per hectare in first to second week of June. So, either of these two insecticide can be used to control this pest. One can be sprayed and other can be used as granules. Stalk borer, Chilo oricillus, flourishes most in waterlogged conditions during post-monsoon period. Larvae feed on leaf sheaths or on midribs and then bore into stalks by making circular holes in the rind. So, these are the infestation symptoms. You can see larvae here. What can be done to control it? Stripping of dry foliage twice in September and October. Release of Trichogramma chilonis at the rate of 50,000 adults per hectare at 10 days interval from July to October. Now, next important insect is internode borer. As the name suggests, it bores into the internode of cane. Chilosacchariphagus indicus larvae damage top tender part of cane when young, and later on it tunnel through upper internode when grown up. So you can see the damage in this picture. Larvae feeds on fibrous tissue by tunneling and tissues turn red. So you can see in this picture, this is tunneling. Tunnel is made by the larvae, and sometimes it can break break also. You can see the internode borer larvae. You can see here also. Stripping of dry leaves during September, October and trash removal at harvest is important practice. Another is release of T. chilonis at the rate of 50,000 adults from July to October at 10 days interval. So this bioagent is quite effective against many insect pests of sugarcane. Number 6 is Gurdaspur borer. Scientific name is Acigona stenialis. Top leaves turn pale and ultimately dry up. In initial stages, larvae feed gregariously in top portion of canes by making spiral galleries, thus killing the plant. After feeding on cane, it move on to new ones. Means one larvae can feed on several canes, and you can see the damage being done on the right side. You can also try to identify the larvae. This is the larvae. So far management is concerned, mechanical removal of infested plants from second week of July. Plowing of stubbles and removal of trash at harvest are good methods of control. You can see the damage being done by Gurdaspur borer, and here also the drying of the shoot can also occur due to infestation of Gurdaspur borer. Next is termite. I think each one of you understand what are the damages and how it do the damage to the sugarcane crop. So scientific name is Odontotermus obesus. Infestation commences with planting of sets, means right from the sowing time. Termites damage cut ends and buds of sets affecting germination adversely. Sets are completely eaten away, leaving only outer hard rind. In attacked plants, outer leaves first show signs of drying, and attacked canes may come off easily if you pull them. Now see the large number of termites are there, and they are eating this cane. You can see the damaged cane. Again damaged cane, they have eaten everything, and your stem or canes are drying. So what can be done to control termite? Use well-rotten manure only. Remove stubbles and debris of previous crops. Dig termitaria to destroy the queen. Means if you destroy the queen, then their life cycle will be finished. Application of chlorpyrifos 20 EC at the rate of 6.25 liter per hectare or chlorantraniliprole 18.5 SC at the rate of 500 to 625 milliliter per hectare over cane sets in furrow at time. Application of chlorpyrifos 20 EC at the rate of 6.25 liter per hectare through irrigation water as post-planting treatment. So both options are available for use of chlorpyrifos, which is quite effective against termite. The rate of application is 6.25 liter per hectare. It can be either applied as foliar spray by dissolving it in say 600 to 800 liter water and dissolving the same amount and then spraying in 1 hectare area. Otherwise, many farmers or intelligent farmers can use it with irrigation water also. White grub, you can see as the name suggests, this grub is quite harmful. It mainly eats the roots, and you can see the orange, the head is orange, rest is white, and it is just like a C-shaped. Many times it is C-shaped insect. So, scientific name is Holotrichia consanguinea. Fleshy grubs are highly destructive to roots and underground stalks. Canes collapse, dry up and become unfit for crushing or juice extraction or planting. Affected clumps often harbor 7 to 10 grown-up grubs in roots. They are called grubs, kind of larvae. They do not have any legs. Now you can see the grubs. These are the grubs damaging stage, and sometimes beetle are also harmful, and you can see the grubs. Small neem trees be sprayed with chlorpyrifos after first summer rain to kill adult beetles which congregate on these trees for feeding. If you are growing sugarcane crop and around that sugarcane crop, if you have neem trees, then you should take care of controlling the beetles, because the beetles, the parents of grub, will like to live on the neem tree. So better to control or kill those beetles so that you do not get any grubs produced. Hand collection of adult beetles, use light traps and soil application of chlorpyrifos at the rate of 10 g at the rate of 25 kg per hectare during first fortnight of June. So chlorpyrifos can be used. Now pyrrhilla is also important insect pest of sugarcane crop. Scientific name is Pyrrhilla purpusilla. It remains in colonies on lower surface of leaves. Both adults and nymphs suck the sap, causing yellowing and drying of leaves. Leaves can turn up pale yellow and shrivel up later. Shrivel up later means this decrease in size, and even canes dry up and die when attack is severe. Insects excrete honeydew, which falls on leaves and act as good medium for black mold. Now black mold growth will be there on this honeydew. So black coating actually interferes with photosynthesis and very little food is manufactured by the plants. So it is harming in two ways. One is direct loss by sucking the sap, and secondly through honeydew it reduces photosynthesis rate of the plant, and many times it has become very severe pest in India. Now you can see the pyrrhilla insect. This is pyrrhilla insect, and you can see how it is producing honeydew, and this is pyrrhilla infested crop. See more pictures of pyrrhilla insect and see sooty mold appearance here. Sooty mold, all sooty mold. So how come these leaves can go for photosynthesis? May not be possible. So there may be less production of dry matter also. Management of pyrrhilla, spraying of imidacloprid, very effective insecticide, 17-8 SL at the rate of only 125 ml per hectare. The dose of this insecticide is quite low. If pest migration is noticed in other crops during pre-monsoon period. Conservation of Epiricania melanoleuca. It is a biocontrol agent. Cocoons during pre-monsoon and redistribution or colonization of 4 to 5 lakh eggs and 4,000 to 5,000 cocoons per hectare during July to August. Now next insect is scale insects. You can see the damage system being done on the right side pictures. It is called as Melanopsis glomerata. Nymph and adult female inside a scale covering feed by sucking juice and cause shriveling up and stunting of the canes. Drying of canes with reduced internodes. You can see on the right side the canes have dried and internode size is also reduced. That means the yield will also go down. How to go for management of this scale insect? Dipping of sat in water with imidacloprid 17.8 SL at the rate of 0.3 ml per liter for 15 minutes. Means you need to prepare the solution of this insecticide and then keep the sets immersed in this water or solution for 15 minutes. Ratoon from infested crops should not be taken, it should be avoided, and spraying the crop with same insecticide imidacloprid 17.8 SL at the rate of 125 ml per hectare or thiamethoxam 75 Sg at the rate of 160 ml per liter in December and January in Exali and in June and July for spring planted crop. So, it gets dominated particularly in spring crop in subtropical belt. Next is black bug. You are seeing on the right side, they are there on the leaves of sugarcane. So, scientific name is Cavallarius sweetie. On young plants, nymph and adults suck cell sap from central hole. Central hole means central leaves. On grown-up plants, they prefer to feed within leaf sheath. Leaves become paler and show holes after feeding. Spray towards holes with imidacloprid 17.8 SL at the rate of 125 ml per hectare and dissolved in 1000 liter of water. Number 12 insect is sugarcane whitefly. You can see or you can identify whitefly on the right side picture. Allurolobus barodensis is the name. Nymph cause suck cell of leaves, means nymphs are important, they suck the cell sap. Yellow streak seen on attacked leaves and crop become pale green. Sugar recovery and yield is reduced, that is really quite negative point. Black mold grows on honeydew, excreted by pest and interferes with proper functioning of the leaves. Now you can see the diseased leaves or leaves that has been infested by whitefly. Management of sugarcane whitefly, what can be done to control? Clipping and disposing of affected leaves prevents spread. Avoid ratooning especially in low-lying and waterlogged areas. Plant repellent plants like peppermint or attractant plants for natural enemies like French bean predatory thrips. Apply chlorpyrifos 20 EC at the rate of 150 gram AI per hectare. AI is active ingredient in 750 ml formulation per hectare. Sugarcane mealybug, Saccharococcus sacchari is the scientific name. Bugs seen clustered at basal nodes of canes and exposed when leaf sheath removed. Nymphs and wingless female adults suck the cell sap. First noticed in appreciable numbers when canes are 4 months old till harvest. Drought affected crop is more severely damaged. They excrete honeydew. A sooty mold develops giving a blackish appearance to canes. Now you can see picture of mealybug. Now what is management of sugarcane mealybug? Grow attractant plants. Coriander attracting wasp, use tolerant varieties, detrashing of sugarcane leaves. Drain excess water and avoid water stress conditions. Conserve and augment natural enemies. For example, Chilocorus, Hyperepsis, Chrysoperla, Aphistis, Cocophagus, Enchiricercia, etc., etc. In severe infestation, spray acephate 75 SP or dimethoate 30 EC at the rate of 2 gram or 2 ml per liter after detrashing. Next insect is sugarcane woolly aphid. Scientific name is Ceratovacuna laningera. Nymph and adult found on lower leaf surface, suck cell sap and honeydew dropped on upper surface of lower leaves. Means upper surface of lower leaves means suppose this is your cane and these are lower leaves. So on the upper surface of lower leaves, this is important to understand. Honeybee encourages fungus growth which results in black coating called sooty mold on upper leaf surface affecting photosynthesis. Sap drainage results in leaf gradually turning yellow and pinkish and ultimately dries. In case of severe infestation, there can be very high losses caused by this insect, which is woolly aphid. Now you can see the damage symptoms by woolly aphid. Here you can see the whitish kind of structures are formed. It looks like wool. Therefore, name is woolly aphid. What can be done under the management of sugarcane woolly aphid? Avoid excess irrigation. Select seed material from pest-free areas and uninfested areas. Destroy affected leaves when pest problem initially noticed in a limited area. Utilize sugarcane varieties less susceptible to the woolly aphid. So the area where its infestation is more, under those conditions, the varieties which are resistant to woolly aphid should be preferred or should be grown. Spray thiamethoxam 25 wg 0.005% or imidacloprid 17.8 SL. The concentration is 0.003%. Acetamiprid 20 SP 0.004%, chlorpyrifos 0.04% or dimethoate 30 EC 0.051%. So there is wider choice of insecticide to control the woolly aphid. So either one of these insecticide can be used to control it, but control should be adopted when there is economic injury level of the insect. Now, you have seen many important insect pests that reduce the quality, the growth and productivity of sugarcane crop. Similarly, there are number of diseases that can affect the sugarcane crop. They can reduce the growth, they can reduce the productivity as well as quality. Many times diseases can reduce the sugar content in the juice, hence the recovery of sugar will be less. So it is long duration vegetatively propagated crop and attacked by number of pathogens. These pathogens can be fungi, bacteria, phytoplasma and viruses from germination till maturity. About 60 diseases have been reported in India to attack this crop. And a dozen diseases are important. So it is not that all the 60 all 12 disease will come in every crop, in every region or in every year. It is not. Under certain year, under certain regional conditions, a few disease may come and one of them or two of them can adapt or can go to very severe infection. Most diseases are seed piece transmissible. Interstate transportation of seed material without any quarantine system spreads the disease. So most important, communication of disease happens through set to set, means planting material can carry many diseases. The first and topmost disease of sugarcane is red rot. Set-borne inoculum is major mode of inoculum transmission. Symptoms start from July, August and found till end of the crop. Third to fourth leaf from top starts withering along margins. Later on, whole crown withers away. At initial stages, purple color develops on nodes of cane which turns to reddish brown later. Midrib lesions are major source of inoculum during cane growing season. So inoculum is actually part of the pathogen that can produce the disease in new part. Colatotrichum falcatum is the fungus that causes red rot disease, and typical symptoms are observed in internodes of stalk after splitting the cane longitudinally. Dull reddening of tissues with white spots are seen inside the cane, which omit alcoholic smell, or this smell can be bad smell. Many a times such kind of smell is unbearable. Red rot affected canes are very sensitive to broken from node. They can break. Infected standing stalks serve as a greater inoculum source at later stages of crop. So you can see yellowing of leaves, these are the initial symptoms, and then you can see death of seedling. If it comes at early stage, there can be death of seedling. Yellowing of third or fourth leaf can also happen from the top. Now you see midrib has become red. Now see stalk showing symptoms inside. So it is all reddening, and it is kind of fermentation has happened, and you see some foul smell will come from such kind of canes, and see the drying of the whole crop. So many times it can go to very high extent, and recently in subtropical belt of our country 0238 variety was ruling, most common variety for last many years, but for last 2-3 years this variety is almost wiped out by this fungus red rot. It is coming everywhere. Now farmers are switching over to some newer varieties or better varieties. So this disease is really most destructive disease of sugarcane. Management: grow resistant or moderately resistant varieties of sugarcane. Any set showing reddening at cut ends or nodal regions should be discarded. Healthy seeds should be planted. Pre-treatment of seed canes in moist hot air, 54 degrees centigrade for 2.5 hours at about 99% humidity. So this kind of hot air treatment is good. It not only removes the fungal inoculum, but it can also remove or kill some viruses and phytoplasma, etc. As soon as disease noticed, affected clump along with root system should be uprooted and burnt. At least secondary infection can be avoided. Bunding of affected field should be done to avoid movement of rain or flood water, because along the water movement, the inoculum can be carried from the diseased plant to the healthy plants or diseased field to the healthy field. Rattoning of diseased crop should be avoided. Diseased crop should be harvested as early as possible. Crop rotation should be followed in affected fields. This is most important part of the management strategy, to adopt crop rotation. So what we normally see that farmers in subtropical belt as well as tropical belt of sugarcane, they are following monoculture kind of farming of sugarcane. They may take first crop, second crop, and again in the same field they can go for sugarcane again and again. So by this way lot of inoculum are getting accumulated in the soil. So the disease incidence may happen quite high under monoculture system. Therefore, it is need of the hour to change rotation. Adoption of at least 2-3 rotations should be professed or should be adopted in those areas. Suppose you are following 3 years crop rotation. So in that case, sugarcane crop should come once in 3 years. So first 3 years it will come one, next 3 years it will come once. So likewise, these kind of rotations can help to reduce or minimize the incidence of red rot of sugarcane. Now next important disease is smut disease. As the name suggests, it is smut. Smut means black. So Sporisorium sitaminum, new sprouts lean and lanky, profuse in number, growing point project out of a long black whip with black spores. So this is whip. This is the whip, and it is full of black spores. Slender and thin canes with erect and pointed leaves. Such plants easily located before production of smut whip. Now you can see black whip formation in smut affected sugarcane. So you can see this very clearly showing symptoms. Students can easily identify and remember the symptoms of smut in sugarcane. See this whip, again whip very clear, and in this also. So this is your smut disease symptom in sugarcane. Now management of smut, resistant or moderately resistant varieties should be used. Healthy seed should be planted. Pre-treatment of seed pieces by dipping in a 0.2% solution of carbendazim helps reduce incidence. So carbendazim is also known as bavestine. One can prepare 0.2% solution easily. Take 2 gram of bavestine per liter of water and make the desirable solution and spray. Removal of affected clumps showing smut whip during tilling phase effectively reduces disease incidence. Next is wilt. It is caused by Fusarium sakari. Symptoms usually appear after the monsoon. Infected clumps individually or collectively show stunting and yellowing of top leaves. In severe cases, whole clump dries and canes become hollow and lighter in weight. Red discoloration in internode is more intense towards nodes which do not emit specific odor. Now you can see the wilted plants of sugarcane. It is complete wilting, drying, and there can be all kind of destruction of this crop. And see how if you break the stem longitudinally, how it looks. It looks like this, but there will not be foul smell like in case of red rot. Shrinkage of cane. See this kind of shrinkage happen in the cane. This is a typical symptom of wilt in sugarcane. Now management of wilt, healthy seed should be planted, means better to go for seed treatment, and seed treatment can be done with 0.2% carbendazim. Take 2 gram carbendazim per liter of water, make the necessary solution as per requirement, and then put these sets, 3 budded sets or 2 budded set, whatever you are using, for at least 15 minutes in this solution. Then crop rotation should be followed in the affected field. So as it was suggested to control or reduce the incidence of red rot by adopting crop rotation, same recommendation is here also. So overall it can be said that crop rotation are not directed to stop or prevent any specific disease. They are useful in preventing almost all the disease. Crop rotations are also useful in reducing the incidence of insect pest also. They are also important for controlling the weeds also. Therefore, value of crop rotations is being ignored by the farmers. But in this case also, if you follow crop rotations, means rotating the crop in 2 years, 3 years or 4 years time, then you will get less incidence of insect pests and of course diseases also. Now, polka boing disease. So, Fusarium molliforming is the causal organism. Polka boing is a Javanese term denoting a malformation or distorted top. So, this is from Java actually. Java is part of Indonesia, and you know in Indonesia sugarcane is grown. So, initially chlorotic patches towards base of young leaves. In acute cases, distortion of stalk with an external and internal cut-like lesions and rotting of apical parts. You can see here. See more symptoms. These are typical symptoms of polka boing disease. And then you can see the blackening or rotting of the shoot part or top part. This can be very dangerous. And top rot, here you can see top rot. So the growth of the top is stopped. That means plants will remain stunted. Now, what is the management of polka boing disease? Spraying of different fungicide like carbendazim 1 gram per liter of water. It will give you 0.1% solution, or Peletox 0.2%, or copper oxychloride 0.3%, or dithane M45 3 gram per liter of water is quite effective. You can choose one of these fungicide and use to control this disease. 2 to 3 sprayings with an interval of 15 days reduce multiplication of pathogen and losses in yield and juice quality. Sat rot or pineapple disease. It is caused by Ceratocystis paradoxa. Sats may rot before germination. Shoots die after reaching a height of 15 to 30 centimeter. Sats dried up, reddish color becomes black with lots of black fungal spores adhering to it. Central portion of shoots becomes red and contents of sat rotting. When split opened, affected sats exhibit pineapple udder. Therefore, the name comes pineapple disease. Now you can see the affected plants, their growth is severely reduced. You can see there is no trilling, and see how the sat is drying, and these are the symptoms when you break open longitudinally. Healthy sats should be obtained from disease-free field. Proper drainage, planting depth should be shallow. So these are the measures to manage this disease. Other measures are field sanitation, sat treatment with fungicide like carbendazim, 0.1% solution. Apply carbendazim 2 gram per liter at root zone area, and same follow at 15 days interval. Next important disease is rust. It is caused by Puccinia arianthi. Earliest symptoms are a small elongated yellowish spots visible on both leaf surfaces. Spots increase in length, turn brown to orange brown or red brown, which coalesced and formed large irregular necrotic areas showing rusty appearance of leaf, and it also results in premature death of the leaves. You can see these are rusty spots on both sides, they can occur, and here the starting point is yellow spots on leaf, and then they can advance red brown spots, and further they can advance to rusty appearance. And you can see it can reduce the photosynthesis also, because greenness of the leaves have vanished. Now, what is the management of rust disease in sugarcane? First option is grow rust resistant varieties. Affected leaves should be removed and burnt immediately. Sugarcane grown in fields receiving recent application of mild mud is typically prone to rust. Spray dithane M45 at the rate of 2 gram per liter for one spraying. Application of triazole or strobilurin or pyraclostrobin fungicide at the rate of 3 gram per liter of water. Seventh number disease or important disease of sugarcane is yellow leaf disease. It is referred as YLD and caused by sugarcane yellow leaf virus. Leaf discoloration happens, stunted growth and bunchy top appearance of the plant. Yellowing of leaf midrib on lower side of leaf, yellowing of midrib on leaves 3 to 6 cm down from the top. Subsequent leaf drying along with the midrib in 3 to 5 leaves from top. Stunted growth of cane, particularly internodes, are affected. Now you see this viral disease, yellow leaf disease. These are the symptoms, and you see drying of leaf. In advanced stage, drying of leaf means it will not have photosynthesis, and then there will be less production of cane and less production of sugar also. So overall, this YLD will affect the quality as well as productivity of the sugarcane crop. What is the management options for yellow leaf disease in sugarcane? Selection of disease-free sets. That is the easiest and best way to prevent the infection of this disease. Field sanitation. use resistant variety. And since it is viral disease, one can go for this hot air treatment or moist hot air treatment. Secondary transmission is by insect vectors. So those vectors should be controlled. To control spread in a standing crop, spray of systemic insecticides to contain or to prevent or to kill aphids may be done. For this, we can use imidacloprid, etc. Now, grassy shoot disease, it is commonly called as GSD, caused by phytoplasma. Initial symptom appears in young crop 3 to 4 months old as thin, papery white young leaves at cane top. Later, white or yellow tillers appear in large number. Below these leaves, profuse tilling. Cane is stunted, means its growth is reduced. Canes are shorter in height. Appears in isolated clumps. Large number of lean and lanky. Lanky means thin. Pale sprouts in clump appear as bunchy grass. Normal stalks are not formed. So, particularly it can go to the ratoon crop. So, now you see grassy shoot disease in sugarcane. Vectors are aphids, can be managed by spraying imidacloprid 17.8 SL at the rate of 125 ml per hectare. This quantity can be dissolved in 600 to 800 liters of water and sprayed onto the crop. You can see this kind of grassy shoot. It has become almost grassy, means the cane development is adversely affected in this. If the cane development is adversely affected, then the cane yield will also be low. The quality, the sugar content will also be reduced. Mosaic, caused by sugarcane mosaic virus. Young leaves of crown held against light source display chlorotic and normal green areas, imparting a mosaic pattern. You can see on the right side picture, this is mosaic pattern. Somewhere you will find yellowness, somewhere greenness. So, these kind of mixing of yellow color and green color may be there. This is called as mosaic symptom. Chlorotic areas show reddening or necrosis. Leaf sheath also displays such symptoms. What can be management? You see mosaic symptoms in the leaf. Seeds should be obtained from disease-free crop. Secondary spread by insects can be controlled by imidacloprid 17.8 SL at the rate of 125 ml per hectare. Next is ratoon stunting disease RSD. Causal organism is Leafsonia xyli, subspecies xyli, a small aerobic bacterium. Affected plants are stunted, being most severe in stubble and ratoon crops. In infected stalks, presence of pinhead-like orange dots of bacteria on internal soft tissue in nodal region. Seeds taken from diseased plants germinate poorly and few shoots that emerge grow very slowly. Treat sets or seeds with hot water at 50 degrees centigrade for 2 hours. Aerated steam therapy eliminates pathogen from infected canes. Use of disinfectants to clean seed cutting tools, which would reduce spread of pathogen from infected to healthy plants. So, what happens? Many times farmers use some implements like gandasa or sickles, etc., to cut the sets during sowing or planting. So, those implements should also be disinfected time to time. Let us study processing of sugarcane. It grows to 3 to 4 meter height and has about 5 centimeter diameter. Stem grows into cane stalk, which when mature constitute about 75% of the entire plant. Mature cane composed of 11 to 16% fiber, 12 to 16% soluble sugar, 2 to 3% non-sugars and 63 to 73% water. This sugar percentage varies from one region to another region, one variety to another variety. Average yield of cane stock is 60 to 70 tons per hectare. Now, sugarcane products and processing byproducts. You can see variety of products are there. The topmost product is sugarcane. Most of the sugar in India and world is derived from sugarcane. The second important crop is sugar beet. Next comes other product like filter muds, your jaggery, etc. Molasses is yet another important product which is received during formation or crystallization of sugar, and this molasses can be used for several purposes. Then juice, of course, juice can be used to make sugar. It can be used to make some food product also, and juice can also be used to sell in the market. Now you will find many people are crushing canes in the cities, urban centers, towns, and they are selling juice of the sugarcane. Now some people are packing the juice of sugarcane and selling in the market. Next product is bagasse. Bagasse, after extraction of juice from the canes, whatever residue is left, that is known as bagasse or khoi. This can be used to produce or generate electricity. It has several purposes. It can be used to make cartons, to make papers, and so on. And then you can have cane tops or top leaves of the cane. They are used as fodder crop. Now, what are the uses of bagasse? You can check. There are a number of uses for bagasse. It can be utilized as a fuel in rural areas. It can be used to generate electricity. It can be used to prepare charcoal briquettes. It can be a producer gas, and it can also give fibrous products like writing paper, particle board, fiber board, bleached pulp, and so on. And there are miscellaneous uses of bagasse. Fresh bagasse can be used for animal feed, poultry litter, xylitol, and furfural. Number of things can be made from sugarcane bagasse. Now, molasses, shira, it can be used for distillery industry where one can make rum, ethyl alcohol, and rectified spirits. So, variety of products, diversified product can be produced from molasses in distillery industry. There are some other fermentation industries where molasses can be used to produce vinegar, sirka, citric acid, lactic acid, and glycerol. Similarly, direct utilization of molasses can be made in animal feed, and also it can be used as a fertilizer. So, you are seeing that very many diversified product can be achieved from sugarcane. Now, sugar processing steps. These are important steps. Students should know them. First of all, what happens? The canes or sugar canes are purchased by the factories, and they are transported from farmers' field to the factories. So, cane receiving and unloading. Farmers bring this cane in bullock carts or in tractor trolleys, in trucks. There are many means of transport through which sugar cane is transported from farmers' field to the industry or to the factory. Then what happens? Cane preparation is done. Here cutting and shredding of the cane is done. Juice extraction by milling and diffusion. Juice clarification is also necessary to remove the mud or impurities, to remove the waxes, and to remove the fibers. Then juice evaporation to concentrate about 65-degree bricks, about 65% sugar you can say. So, water is evaporated here. Then syrup clarification. Syrup clarification remove suspended solids, mud, waxes, fibers, etc. Then centrifugation. In centrifugation, separation of the sugar crystals from mother liquor. And then sugar is dried and packaging and delivering. So, these are important or major steps in sugar production. However, in certain factories or industries, they may be modified, they may be changed. Now, bagasse processing, it is very important part. Pulpy fibrous material left after extracting juice. It contains cellulose 45 to 55%, hemicellulose 20 to 25%, lignin 18 to 24%, ash is 1 to 4%, and waxes less than 1%. So it is really rich in carbohydrates, or particularly cellulose and hemicellulose. Ethanol can be obtained either by acid or enzymatic hydrolysis. Hydrolysis gives hexose and pentoses, which further decomposes to yield ethanol by fermentation. Molasses processing. You see picture of the molasses on the right side. Molasses is final effluent obtained in sugar preparation by repeated crystallization particularly during the centrifugation. The sugar is separated from the molasses. The solid part is sugar and the liquid part is molasses. It is residual syrup from which no sugar can be crystallized. Many products of economic importance can be prepared or produced from it after the processing. It can be used in animal feed also. Now see what can be done to the molasses. So molasses, one can use yeast. Yeast can be used, these are kind of fungi. They can be used for fermentation, and then after fermentation distillation can be made, and then you can prepare rectified spirit, and then redistillation can be done, and then redistilled alcohol. There are many procedures to produce alcohol and rum and other kind of drinks from molasses. Now pressed mud cake is also important byproduct of sugarcane factories. So it is soft, spongy, amorphous, dark brown material containing sugars, fiber, coagulated colloids, which includes wax, albuminoids, inorganic salts. In sugar manufacturing, impurities of cane juice are precipitated either through sulfitation or carbonation process. There are two different processes. In this case, sulfitation is important one for the farmer because it increases sulfur content in pressed mud. Pressed mud is a rich source of organic carbon. Sufficient quantity of carbon is present, and it contains a good proportion of nitrogen, phosphorus, calcium, iron and manganese. So many macro and micronutrients are present in this pressed mud. So it is a kind of organic manure. Now following are the main pressed mud based industries. Use of pressed mud as fertilizer. So many times farmers use pressed mud as a manure, and since it contains nutrient. Pressed mud can be used as an animal feed also with some modifications. Cane wax from pressed mud can be prepared. Biogas from pressed mud can be produced. Steam generation from pressed mud cake, and use of pressed mud cake in building material. From agricultural point of view, it can be composted and it can be used as a manure to supply nutrients. It is excellent source of nutrient and organic carbon. Dear students, hope you enjoyed this lecture very much. Thank you so much.